Step forward in cancer reversal. Korean scientists develop system for focused intervention at transition point of tumorigenesis. REVERT can be applied broadly to cell fate transitions in differentiation, development, and cancer cell reprogramming. Cancer research has come a long way from killing malignant tumors or stopping the advance of the disease to entirely reversing the state of the cell into normal ones, as illustrated by a breakthrough in a Korean lab last year. Building on this work on cancer cell reversal by Korean scientist Kwang-Hyun Cho, a research team led by him has now identified a switch, a more precise point for such an intervention. According to their new report published in the journal Advanced Science, cell fate changes involve abrupt transitions or critical transitions, particularly in tumorigenesis. Understanding the molecular regulatory networks underlying these transitions may enable cancer reversion, reprogramming cancer cells to normal states, the researchers from Korea Advanced Institute of Science and Technology stated in the report. Early studies relied on static analyses, limiting insights into dynamic transitions. Advances in single-cell transcriptomics and pseudotime analysis now help identify transition genes and model regulatory networks. However, tumorigenesis remains difficult to model due to genetic alterations. Focusing on tipping points rather than entire trajectories may better capture key transitions. Recent research highlights attractor states and gene expression heterogeneity, crucial for understanding tumorigenesis and potential therapeutic interventions. The team developed REVERT, Reverse Transition, a novel systems framework designed to identify reversion switches in tumorigenesis by analyzing the attractor landscape of dynamic gene regulatory networks, GRN. Using single-cell transcriptomic data from cancer patients, REVERT employs Boolean network modeling to map the transition from normal to cancer cell states. Attractor landscape analysis is a computational technique used to model and visualize the stable states, or attractors, that a biological system can adopt. In cancer research, this method helps scientists understand critical transitions between normal and malignant cellular states. By mapping these transitions, researchers can pinpoint molecular events that drive tumor progression or, potentially, tumor reversion. REVERT enables the identification of key transcription factors, TF, that can reverse cancer by minimizing a malignancy score. It reconstructs dynamic GRN models within tumor transition states, capturing the stochastic nature of cell state changes. It aims to predict optimal intervention targets for reversing cancer progression and can be applied broadly to cell fate transitions in differentiation, development, and cancer cell reprogramming. The REVERT framework consists of 4 key steps. Identification of the transition state. REVERT clusters tumor and normal cells to define a transition state. This analysis helps distinguish normal-like, tumor-like, and transitional subclones. Dynamic network model reconstruction. A pseudotime analysis tracks differentially expressed genes along the transition from normal to tumor states. By integrating these temporal changes with known gene regulatory interactions, Boolean logic functions are constructed to model transcription factor dynamics within strongly connected components. Cancer score, CS computation. REVERT introduces a cancer score to quantify attractor landscape stability, measuring the relative dominance of normal and tumor cell states. A lower CS indicates a state closer to a normal phenotype. Target identification for cancer reversion. REVERT predicts key transcription factors whose modulation reduces CS, guiding therapeutic interventions. By integrating computational modeling with experimental validation, REVERT provides a powerful tool for identifying cancer reversion targets and advancing precision oncology. Developing a systems framework for controlling cell states based on omics data, compilation of data resulting from the study of various omes of an organism, such as genome, transcriptome, proteome, metabolome, and epigenome, has been a significant challenge in various biological fields, including drug discovery, stem cell engineering, and regenerative medicine. This study successfully leverages this dataset. Future directions for REVERT include integrating multi-omic single-cell sequencing data to refine tumor transition state identification. By combining genomic and transcriptomic information, REVERT can offer even greater insights into molecular mechanisms underlying tumor progression and potential therapeutic interventions for cancer reversion.
Professor Kwang-Hyun Cho highlighted the importance of this discovery, stating, "The fact that cancer cells can be converted back to normal cells is an astonishing phenomenon. This study proves that such reversion can be systematically induced." He further added, "This research introduces the novel concept of reversible cancer therapy by reverting cancer cells to normal cells. It also lays the foundation for identifying precise targets for cancer reversion through the systematic analysis of normal cell differentiation trajectories." The findings from this study hold promise for developing innovative cancer therapies that could potentially be applied to other types of cancer as well. As research continues, this breakthrough could revolutionize the way we approach one of the world's deadliest diseases.